Hello everyone. So today what we're going to be doing is pulling some data from the short read archive. This is going to be our first endeavor into Galaxy. And so just to give everybody a brief overview of what we'll be doing, after we pull some data that we were looking at in the last video, we're going to be uploading it into Galaxy. We're also going to be finding which reference genomes make sense to map the data that we're going to be downloading. And then afterwards, we're also going to be using a visualization tool called Integrated Genomics Viewer. This is going to be a pretty quick summary of what we're going to be doing across the different RNA-seq data sets that we're going to be exploring, with the idea that if we understand what the depositors or what the authors of the data sets, what their notes are in the short read archive abstracts, then we can have an idea of what we expect to see in their data, and we can do some cool stuff afterwards. So the first thing that we'll do is just jump into Safari, and we're going to need to download the data that we're going to be using. So we're going to go to the short read archive, so SRA, curious about what SRA churros are, but that may be a different video. So SRA and CDI, that should get us to where we need to be. First thing we're going to do is type in our search phrase. So remember, this was HIV -1 and RNA -seq, because that's the proper way of saying RNA-seq. Previously, I was using quotations between the main terms, and I added an operator "and", but the default setting for the search algorithm will see me not including "and", or what will work without me including "and". So looking there, what we have is our 2551, so this hasn't changed since we started looking at it. And this should be the same type of, or this should be the same experiment that we were starting out with. Just jumping in real quick. No, this is a different, this is a slightly different experiment than what we were looking at before. So I remember, and I wanted to use this this first sample because it sounded a little bit interesting. I remember the previous data set that we ended with had something to do with Kaposi sarcoma and HIV. So I'm just going to add that to see if we can actually find this. And yeah, so we found some sequences that are from Kaposi sarcoma patients. I don't remember which one was the one that we were using exactly. But I did want to just check to make sure it had something to do with endemic versus epidemic Kaposi sarcoma. So it does seem like this is the one that we were looking at previously, and we can just start out with this. So what we're going to do is we're going to need to find out how to download that data set, the individual data set. And so if we just scroll down, we can actually see that this is the run. So we can pick out this, click on it, and then this goes, we can make this a little bit bigger. This takes us to being able to actually download the data. And we have some other notes that we can look at in the near future. What we're going to be doing is going to data access, and then if we wanted to download the run, it would be here. But this would give it to us, from what I understand, in a not in FASTQ. This would give it to us, from what I remember, in a compressed format, which if you're not sure, you can always just try. So what we can do is we can just click and see what it winds up being. I'm pretty sure that this is not going to be in the FASTQ format. which is what is most easy for us to use in Galaxy. So if we click on reads, this gives us a chance to look at individual reads, just some examples. Analysis, this gives us an idea of what percentage of what percentage of reads are from different animals. So we would expect human RNA-seq reads, it would be predominantly human reads, which is actually what we see here. And interestingly, very few of these reads actually map to, or at least in this superficial analysis, very few of these reads actually map to any viral sequences. It also seems like we don't have that many reads contaminated from other, from bacteria, for example, or other human pathogens, and this is good to know. So we can go to download and FASTA/FASTQ. Actually, let's go back. So we want to be able to download data from this. So this is the SRR number. Remember, we have a bunch of different IDs, so I honestly don't remember which ID off the top of my head, so I have to do some troubleshooting whenever I do this. So let's just type in this SRR number, and then we can show the runs. So this SRR number isn't the right ID number that we'll need, so we can just go back to that main page that gives us a bunch of things. So this is our main page. We'll come back here. So we can click. FASTA/FASTQ, and we can ask for experiments, and then so they also try to help you out here. So we can click this. You can enter the experiment accession or a comma-separated list of experiment accessions. So we need to look for the experiment accession. Sometimes it gives a hyperlink to explain what that actually means a little bit more. So they give you these SRR numbers. So in theory, SRR should be working, but sometimes they ask you for this experiment number. So there are reasons why they subdivide this because of things that I'm not super sure of, just to make our lives less easy, right? So anyways, let's try this accession number. And then, oh, okay, so this gives us something. So, okay, we can click that. No, I don't want to do that. It's like, okay, oh, yes. So this is selected from this experiment, and this just gives us our confirmation. This was the number that we were looking at before. So it gives us some different download options. So we can be filtered by, let's say, certain quality scores, things like that, which we'll talk about a little bit later. You can clip it, for example, if you start doing quality control and you don't want the ends because, let's say, the quality of the bases towards a given end may be problematic, you can clip it a little bit. Frankly, most modern mapping software will handle this, so you don't necessarily need to do a lot of data cleanup, and that actually winds up simplifying data processing and the analyses. But then there are some potential caveats to this approach, which we'll talk about a little bit later. So FASTA and FASTQ, we've already seen FASTA. FASTA is basically, we can jump into one of these examples here, where, oops, nope, I don't want to use that program. I just want to use this text editor because it is simple. Just go, I don't know if Notes isn't. No, Notes isn't. Notes isn't. I have a problem with that probably. Okay, text edit should be decent. But yeah, so the anatomy of a FASTA file, FS or FASTA, is just basically a name line where you have this greater than symbol and then stuff afterwards. That's the name of the following sequence. And then you have the following sequence in either nucleobase space, which is ACGT, or AUGT, or what is it? ACGT if it's DNA, or if it was RNA that was handled in a bioinformatic way, or ACGU if you're just trying to look at it from an RNA perspective. But what we see here is this is just the rest of the sequence. And then in this particular FASTA file, it's a single FASTA file. You can also have multiple FASTA files or multi-FASTA files where you just have many different files like this, where you have name, stuff in the middle, which corresponds to whatever the sequence is, and then you can have it concatenated or just following immediately name, stuff in the middle, name, stuff in the middle. FASTQ is a little bit different, and we'll see that as soon as we download our example data set. So we're going to hit FASTQ here. We're going to download. This is kind of a big data set, so it's going to take a little bit of time. So don't be discouraged. Whenever we continue to work in the space, I would encourage everyone to get rid of as much get rid of as many files as you're not using as possible, because it will make data management a little bit easier. What I want to avoid is people having terabytes of space on their computers just clogged up. So what we'll be doing is downloading small data sets, processing them on Galaxy, summarizing them, and then y'all will be sending me the results for that, and then you'll be able to wipe your machines, and you'll be able to do the next set and the next set and the next set. Galaxy also has a way of publishing results in an intermediate way, where you can actually push your results to be explorable on Galaxy, and it doesn't wind up using your quota space, which is nice. And then depending on how that is done, it could also potentially allow you to permanently cite that in the future. But we'll talk about some of the cooler things that Galaxy can do besides data analysis. But just to emphasize again, like I don't want y'all's computers to be clogged up with files, because that winds up being a real limiting factor for this type of work. So if you remember from one of our earlier videos, there's a little bit less than 5 terabytes of raw data that we're going to be going through. Each analysis step is going to be adding about that much, I guess, space onto a given computer. To give you an idea of what the data sets are. So if you have a data set that has, for example, a lot of sequences, so like a FASTA or a FASTQ format or multi-FASTQ file, if you map that data that is basically a collection of strings becomes a table, which has roughly as many entries as the number of sequences that you're trying to map. So those files get very big very quickly, and depending on what you're doing with them, you may have to do a couple of different steps. So just estimating, if we're starting with 5 terabytes, if we do mapping, we're going to approximately double that just with the map summaries or the BAM files. So that bumps it automatically up to 10 terabases. Yeah, so it gets tricky. And if we wanted to do, or if we wanted to use different tools, we would actually wind up doubling it even more. So let's say if we wanted to evaluate 2 different mapping tools, and that's all we were doing, which is pretty simple. You would start with 5 terabytes, 5 terabytes of data. Each tool would add roughly about that much file space to your machine, and you would wind up with 15 terabytes, of space used up. So we are going to try to minimize that as much as possible. Yeah, so I'm going to let this finish downloading. What we can do is also check the file that was downloaded previously. Remember, this was the SRR. So remember, whenever I downloaded that, I didn't specify what file format I wanted. So by default, these are compressed in a proprietary way that the SRA tools package, which is something that we haven't talked about explicitly, but it's a package that um, allows you to decompress these data files. But if you tried to open it, for example, in a text editor, it's going to give you some garbage that's like half interpretable, half not. And it's because it's a kind of compressed file that um, that you need a special tool to be able to open. Um, because we are all learning, and because I want to focus on paying attention to moving individual files, um, and like I really want y'all to actually see where the files are going in these different parts. I want like I don't want to jump into using the SRA tools, but once you become proficient, the SRA tools can be really useful, just because it allows you to download multiple data sets at once. Um, Even though sometimes it does take a while, even if you're using that tool to download the data sets. Um, but yeah. So this is still going. It is maybe like 25% done, and I'm just going off of this SRR file size because, again, this is compressed. So I'm thinking that this may be a couple of gigs, and it just needs to go for a little bit. So um, we are going to pause, I think, let this finish, and we will start up shortly. So stay tuned.